Hello, hello. I'm here to talk to you about your DNA molecule. Isn't it beautiful? You had no idea that you were such a wonderful molecular biochemist. Making DNA, making it happen, having fun. All right, so when we talk about this DNA molecule, there are a few things that I want you to remember about the structure and how it relates to the function of DNA. Okay, we can break this DNA double helix into a couple different ways. One way, we can look at each individual nucleotide. What is a nucleotide? Yeah, ignore everything under my hands. We're looking at this. What is a nucleotide? This is the monomer, or the building block of our nucleic acid. This nucleotide is a guanine nucleotide, okay? What does a nucleotide consist of? Well, it's got a phosphate group, our lovely pink little molecule up there. It also has a deoxyribose sugar, okay? The phosphate and the deoxyribose sugar are the ones that incorporate in the backbone. And then we have our nitrogenous base, guanine, okay? So these are the three parts that make up a nucleotide. These are all bonded together with covalent bonds. And if you recall from chapter, I don't know, like a long time ago, covalent bonds are the strongest type of chemical bonds that we have. So looking at our nucleotide, one nucleotide, and then as we look at our giant macromolecule, you can see individual nucleotides, right? There's one, there's another one, there's another one. These nucleotides, monomers, are put together to make our macromolecule. But we can also dissect this molecule another way. Okay, if we're looking at it as a ladder, and I described it as a twisted ladder because it, normally it's looking like this, but that's hard with our paper molecule. Looking at our ladder, we've got the sides, the vertical parts of our molecule. Okay, we describe this as the backbone of the DNA, and we're looking at it's just covalently bonded deoxyribose sugar phosphate, deoxyribose sugar phosphate. All right, and here's really interesting. Do you see how it kind of points this way, right, with that peak of that pentagon? Okay, that's showing you the 3' to the 5' end. Now, you were probably surprised, and I saw the struggle some of you had when you were putting this molecule together, that when we look over here, oh, it's upside down. Oh, that's by design. And we will talk more about how that is appropriate and helpful with lesson 3, okay, when we're talking about replication, but it's supposed to be that way. All right, so when we look at it, the backbone on this side, you see that peak, that point is pointing the other way. Okay, now the rungs of our ladder are nucleic acid, our nucleotides. So many words, right? So many words. Did it again. Our nitrogenous bases, that's the N word that I'm looking for. Nitrogenous bases, not nucleotide, that is our 3-part, right? Not nucleic acid, which is our whole macromolecule. Nitrogenous bases, we've got 4 of them. 4 of them, adenine, thymine, cytosine, and guanine, okay? Adenine and thymine are always paired together. Adenine and thymine are always paired together, and you can remember that because apples are in a tree. Apples in a tree, A and T, okay? Guanine and cytosine are always together. Cars in the garage, cars in the garage. So we've got apples in our trees and cars in the garage. That's how we can remember which nitrogenous bases pair with the other. Now, as you look at this, it's in a random order, or so it seems, right? There is no right or wrong way to put these nitrogenous bases together. This is actually where the coding comes from, how the genetic information is stored in the order of the nitrogenous bases. We'll talk more about that in chapter 13, but do know it's coming, okay? Other questions about this? Oh, yeah. Remember how I said covalent bonds between our nitrogenous base and our backbone, our deoxyribose, and then covalent bonds between our deoxyribose and phosphates? Here's where it gets really interesting, because these nitrogenous bases are not covalently bonded. Say, what? They're held together by hydrogen bonds. Ooh, that doesn't sound very strong because they're not. Hydrogen bonds, recall, are some of the weakest bonds. They're stronger than van der Waals forces, but They are weaker than ionic bonds and weaker even more than covalent bonds. And that's very important when we talk about 12-3. So, covalent bonding, hydrogen bonding. I think that'll do it. Yeah, let me know if you have any questions about our deoxyribonucleic acid macromolecule. Yay, we love science. Okay, bye. Technology.